Welcome back to the Deep Dive. Today we're tackling something pretty fundamental, a new finding that, well, it really challenges a core idea in biology, how traits get passed down.
Right. The assumption has always been it's all sealed in the DNA sequence, in the nucleus.
Exactly.
Yeah.
But we're diving into a mechanism where traits seem to travel across generations without touching the genome itself.
This is all about epigenetic inheritance, specifically how signs of longevity, markers for a longer life, are transmitted.
And our sources, they detail some amazing research on roundworms, right?
Duh.
C. elegans.
That's the one. Tiny worms. But they're revealing this unexpected messenger service running between the body cells and the reproductive cells.
So our mission today is to unpack this. We'll look at the surprising roles of, well, lysosomes.
Usually just seen as recycling centers.
Right. And histones, the proteins that package DNA. We want to trace how a change in a parent, maybe even from something like diet, can set the lifespan for potentially their great-grandkids.
It really forces you to rethink what hereditary information even means. So yeah, let's jump into the research. It starts with a longevity puzzle.
Okay, let's set the scene. The Wang lab. They were looking into aging in these C. elegans worms.
Their first goal was pretty standard aging research. How to make the worms live longer.
And they did it by tweaking the worms genetically.
Yeah, they engineered them to produce more of a specific enzyme inside the lysosomes.
Those cellular recycling centers we mentioned.
Exactly. And that initial step worked really well.
How well?
Dramatically well. Boosting that enzyme gave the worms up to a 60% longer lifespan, which, on its own, that's a big deal in aging research.
Okay, proof that tuning lysosomes impacts longevity. Solid finding.
But then came the twist, the part that shifted everything.
This is where the inheritance part comes in.
Precisely. They took these engineered, long-lived worms and crossed them with what are called wild-type worms.
Meaning just regular worms, the ones without the genetic boost for that enzyme.
Exactly. Standard lifespan, normal genetics for this trait.
So, logically, you'd think the offspring wouldn't get the long life benefit if they didn't inherit that specific gene modification.
That's what standard genetics would predict, yes. The source of the extra longevity, that specific gene change, wasn't passed on to all of them.
But that's not what they saw.
Not at all. The offspring, even the ones that were genetically wild type, still lived significantly longer than normal.
Wow. So they inherited the memory of being long-lived without the actual gene for it.
That's a great way to put it, a memory of longevity. And it wasn't just a one-off fluke.
How long did it last?
This longevity boost, this marker, was passed down for up to 4 generations.
4? That's quite persistent for something not written in the DNA sequence.
It's truly astounding. And that persistence is what directly challenges the traditional view, that only the DNA in the nucleus carries heritable information.
So we've got this clear 4-generation effect. Something non-genetic is definitely being inherited, which begs the question, what is it? What's being passed down?
Right. And how? This is where we have to look beyond the genome to the epigenome. The how involves this fascinating bridge they uncovered.
A bridge between what?
Between changes in the worm's body, its somatic cells, and its reproductive cells, the germline, eggs and sperm.
Ah, the Weismann barrier.
Yeah.
That old idea that information doesn't flow from the body to the reproductive cells.
Exactly. That barrier has always been seen as, well, pretty much absolute. Information flows out from the germline, but not back in. This finding really pokes holes in that.
So if the worm's body figured out how to live longer, how did it tell the eggs or sperm to pass that ability on? Let's get into that mechanism. You mentioned lysosomes earlier. More than just garbage disposal.
Absolutely. We're learning lysosomes are much more sophisticated. They're like central command hubs for sensing nutrients and stress.
So they feel out the cell's condition.
Yeah, they monitor what's going on, food levels, cellular stress, and then they send signals. They communicate.
Okay, so the optimized lysosomes in those long-lived worms were sending out longevity signals. But how does that signal get from a body cell all the way to the germline? That seems like a huge leap.
It is. And the messengers making that leap, according to this research, are histones.
Histones. Yeah, okay. I usually think of them like spools for DNA. They help pack it up tightly or keep it loose so genes can be read.
That's the classic view, and it's correct. They organize DNA. But here, they're taking on an extra job, carrying information across cellular boundaries.
So they're not just structure, they're also signals. How does that work? How do they travel?
Well, the pathway they mapped out is quite specific. First, those positive changes in the lysosomes from the enzyme boost,
Uh-huh.
they trigger other processes inside the cell, and this leads to an increase in a particular type of histone, a specific variant.
So more of this specific histone variant gets made. That's the carrier molecule. But how does it physically get into the egg or sperm cells? Isn't that barrier supposed to stop things like proteins moving across?
This is where it gets really clever, almost sneaky. The histone is actively transported from the body tissues into the germline.
Actively transported. By what?
It seems to hitch a ride. It uses proteins whose normal job is to deliver nutrients like fats and other resources to the developing eggs.
Wow. So the body basically hijacks its own nutrient delivery system to send these epigenetic messages.
It appears so. It's repurposing existing pathways for this new kind of information transfer. Incredible efficiency.
But is it just sending the histone over? Is that enough?
Good question. It's not just about getting the histone there. Once it arrives in the germline, it undergoes a change. It gets modified
Modified how?
chemically. Specific chemical tags are added to it. And this modification is the key epigenetic part. It doesn't change the DNA sequence, but it changes how the genes nearby are used or expressed.
Okay, so the histone is the delivery truck, and the chemical tag is the delivery instruction telling the genes what to do.
Precisely. In this case, the modification seems to act like a switch, or maybe a dimmer, making it easier for longevity-related genes to be turned on or expressed more effectively in the next generation.
And did they see this happening? See the modified histones?
Yes, they found that one specific type of histone modification was way higher in the germline of those long-lived worms compared to normal worms. It really nailed down the mechanism.
Okay, that connects the dots from the enzyme to the histone to the germline. But this was all triggered by genetic engineering initially. How does this relate to, normal life? Does this happen naturally?
Great question. The researchers pushed beyond the genetic manipulation. They asked, what natural situation might mimic the effects of boosting that lysosomal enzyme?
And what did they find?
Something really fundamental.
Fasting.
Fasting, like not eating for a period.
Exactly, just restricting nutrients.
Yeah.
They showed that this entire pathway from the lysosome sensing the change to making more of that histone variant, transporting it and modifying it in the germline, it all gets switched on when the worms fast. Wow, so there's the direct link. A physiological state, scarcity, triggers this transgenerational epigenetic signal.
Precisely. The parent's experience of nutrient scarcity gets encoded, in a way, via these histone messengers, potentially preparing the offspring for a world where resources might also be limited, maybe by promoting durability.
That's profound. The body captures an environmental signal and sends instructions to its descendants. It changes how we think about inheritance, making it seem much more dynamic.
It does. We tend to think of inheritance as fixed at conception. This shows the parent's current physiological state can actively shape the epigenetic landscape for the next generation.
It reminds me of that quote from Meng Wang, the lead researcher. She said something like, "We always think inheritance is just in the nucleus."
Right.
But now they show this histone carrying information from one place to another, crossing these boundaries.
It's the physical how. How information gathered by the body, the soma, can actually influence the germline. The histone carries this memorable genetic information for generations, as they put it.
Okay, but it lasts 4 generations, then fades. Why does it stop? Why not permanent?
That likely speaks to the nature of epigenetics itself. These tags, these modifications, they aren't as permanent as DNA changes. They're designed to be more flexible, responsive.
So like a temporary adaptation.
Kind of. If the environmental pressure, the fasting or scarcity goes away for a few generations, the cellular machinery might just stop maintaining that specific histone modification. The system resets, ready for new signals. It's adaptation, not evolution in the permanent sense.
That makes sense. An adaptive timer. Okay, let's zoom out. Beyond these worms, what's the bigger picture? Why does this matter for, say, us?
Well, the implications could be huge, especially for understanding health and disease across generations. We already know epigenetics plays a big role in how organisms deal with stress.
Like diet or toxins or even psychological stress.
Exactly. Think about things like the Dutch famine studies. We saw that children born to mothers who experienced severe malnutrition during the famine had different long-term health outcomes, like higher rates of metabolic issues.
Right, we saw the effect but didn't fully grasp the how.
Precisely. We had the correlation, but the mechanism was murky. This research provides a potential molecular pathway. It shows how a parent's metabolic state, may be altered by severe diet or exposure, could transmit signals to the next generation that affect their health risks later in life.
Things like obesity or diabetes risk could be influenced by a parent's or even grandparent's environment.
It opens up that possibility, yes. It suggests our health isn't just about our own genes and lifestyle, but might also carry echoes of previous generations' experiences written in these epigenetic marks.
So our metabolic choices today might be writing instructions for our grandkids' biology. That's quite a thought.
It connects everything nutrient sensing, aging, environment into this long transgenerational story.
Okay, so let's try and wrap up the core findings from this deep dive. We've seen that longevity, or at least markers for it, can be passed down without changing DNA.
That's the headline, yes. Non-genetic inheritance.
And the mechanism involves lysosomes acting as sensors, particularly responding to states like fasting.
Right. They signal to produce specific histone variants.
These histones then travel, crossing that supposed barrier from body cells to reproductive cells.
Piggybacking on nutrient transporters.
And once they arrive, they get epigenetically modified, tagged with instructions that influence gene expression related to lifespan in the following generations. For about 4 generations in this case.
You've got it. It's a really elegant, complex system linking the parent's environment and physiology directly to the offspring's potential biology.
Which leaves us, and you listening, with a final thought to chew on. We now have this verified mechanism where the body can pass on information about stress, adaptation, longevity via these histone messengers. So what other environmental signals? Maybe less dramatic than fasting, things we experience daily.
Like chronic low-grade inflammation,
Right.
or maybe constant exposure to certain common chemicals.
Exactly. Could those subtle, continuous signals also be using pathways like this? Could they be unintentionally setting the biological stage, maybe for better, maybe for worse, for the generations that come after us?
It definitely makes you think about the long reach of our environment and lifestyle, something to consider. Thanks for joining us for this deep dive.